Okay. Good morning, everyone. Good morning or good afternoon, wherever you are. Welcome to the webinar on sustainability, where we're going to be discussing PFAS and related problems. So this PFAS and related problems. So this webinar is part of the webinar series organized out of the IOP Publishing and the new journal and titled Sustainability, Science and Technology. I'm editor-in-chief of that journal. And the journal is open access journal dedicated effectively to all aspects of sustainability in science, technology, engineering, and so on. And we really are new journal. We're looking for publications. I think that our journal has several advantages. So we are, very inclusive, very interdisciplinary. If you look at our website, you will see that we cover many facets of sustainability, and essentially we're operating under the sustainable developmental goals of the UN. It is truly community-led journal, so we have very nice, very diverse editorial board. I really enjoy working with them. That provides really an equitable platform for dissemination of all types of sustainability research. It's a society-owned, so it's IOP owned, so it's not a private per se, and it goes, publication fees that cover goes towards effectively public and scientific good. So I encourage you to explore this journal and see the website for submission formats and special issues and so on and so on. Or reach out to me or editorial board if you have some unexpected sort of article suggestions or review article suggestions in mind. So today we're going to have a very nice panel of presenters, excluding me. I'm just here to interact and engage everybody. I'm Jonas Baltrušaitis, editor-in-chief of the journal. Now, the key people, of course, are webinar panelists. So Sarah, Linda, Clinton, and Jude. They are very important people with lots of depth and understanding about PFAS. So colleagues from USDA, and Linda is from Purdue University here in the United States. So we will cover a bunch of topics. So we start with sources, sort of classifying sources and toxicity with Sarah. Then Linda is going to go in depth into transformations and fate and transport of these materials. Clinton is going to talk now about mitigation, remediation, and risks after Linda, that is. And then Jude is going to finish up with sort of biotechnology advances related to PFAS. So each of them are going to go for 10 minutes. Because of that, because it's a dense topic, we will not have time for questions in between, but we're going to have 10-minute question session afterwards. In other words, if you have a question, submit a question at any time on this platform, and we have colleagues who are going to gather those questions, and they're going to forward them to me, and I'm gonna ask those questions. I'm going to address the speaker at the end of the final session. So if everyone is ready, one more time I want to introduce Sarah. So Sarah Lupton from USDA Agricultural Research Service. She's going to be talking about PFAS classes, sources, and toxicity. Go ahead, Sarah, take it away.
All right, thank you, Jonas. Can you hear me okay? Great. So I'm going to be talking about PFAS classes, sources, and toxicity. A little bit of background: I'm a research chemist with the Food Animal Metabolism Research Unit in Fargo, North Dakota. part of the USDA Agricultural Research Service, and we've been studying PFAS in food animals for over a decade now. But I'm going to be focused on the classes, sources, and toxicity, so some background information on PFAS. So just an overview, I'm going to be covering the classes, polymer versus non-polymer, the sources, uses, products, contaminated sites, focusing on the US, and then the PFAS sources and exposure cycle, including in that PFAS toxicity, general health effects, and then human exposures and human health effects. So PFAS classes, so PFAS, or better known as per and polyfluoroalkyl substances, there are estimated over 14,000 different PFAS compounds depending on how you define a PFAS compound. And within that includes non-polymers and polymers. So you have the perfluoroalkyl substances, so fully fluorinated. You have the polyfluoroalkyl substances that are partially fluorinated, and then you have the polymer substances, which have 3 different groups. So breaking it down further, under polymers, we have fluoropolymers such as PTFE, which probably is one of the most commonly known fluoropolymers, and then PVDF. On the right is an image of the monomers that are used to make the different fluoropolymers that are available on the market. The next class under polymers is the polymeric perfluoropolyethers, or PFPs. They are carbon with a carbon-oxygen backbone with fluorine attached to the carbons. And then you have the side chain fluorinated polymers. These are non-fluorinated backbones with fluorinated branching side chains. These include your fluorinated urethane polymers, fluorinated acrylates and methacrylates. And some of these polymers can be precursors for the perfluoroalkyl acids, specifically the side chain fluorinated polymers. And then once you move into the non-polymers, the kind of the different classes expands quite a bit wider. So under the polyfluoroalkyl substances, so these are the non-fully fluorinated substances. You have the fluorotelmer substances, which includes like the alcohols, the fluorotelcarboxylic acids, the fluorotelmer sulfonic acids. These are going to be a partially fluorinated carbon chain along with some carbons that are hydrogen bonded, and then a functional group at the end. And then you have the polyfluoroalkane sulfonamidos. These include common compounds like atfosa, netfosa, and these are going to have a sulfonate in the linkage along. And then the next set of compounds are the polyfluoroalkyl ether acids, and this includes the compound DANA. These are going to have ether linkages within the carbon backbone. And then you have the polyfluoroalkyl carboxylic acids, and then the chloral polyfluoroalkyl ether acids. So there's some compounds that have other halogenated elements on them. And these, the compounds circled in blue are going to be potential perfluorocarboxylic acid precursors. And then the compounds circled in orange are going to be potential perfluorosulfonic acid and perfluorocarboxylic acid precursors. And further expansion, we get into the perfluoroalkyl substances, so the fully fluorinated carbon chains. These include the perfluoroalkyl acids, which include the perfluoroalkyl carboxylic acids, and in this group includes PFOA, and then perfluoroalkyl sulfonic acids, and of course in this group includes PFAS, which is one of the most detected compounds. And these are what we're going to call like the terminal PFAS, where they don't degrade, they don't have, they accumulate in the environment. These can be produced from some of the precursor compounds that were mentioned before, but also in here are other compounds such as the perfluoroalkane sulfonic acids, which can be potential sulfonate precursors, and then but there's a bunch of other classes of compounds that aren't as widely detected as the carboxylic acids and sulfonic acids. Moving on, we have the perfluoroalkyl ether acids. This includes a compound HFPODA, which is Gen X. That's a common precursor compound. And then the perfluoroalkane sulfonamides, so the FOSA. And these compounds are also precursors. Some are precursors for only the carboxylic acids, but some are precursors or potential precursors for both the sulfonic acids and the carboxylic acids. All right, so moving into PFAS production background. PFAS compounds have been commercial scale production since the 1950s. As I said before, two of the main compounds that we find a high occurrence of is PFOA and PFAS. They have unique physical and chemical properties. They're oil, water, stain, and soil repellent. They have chemical and temperature resistance, friction reduction, and also surfactant properties. Due to this, they're in many compounds that are used for health, safety, and modern life. Looking at the structures on the right, a lot of these repellent properties and surfactant properties come as a result of the hydrophobic and hydrophilic portions of the molecules. So when we look at polymer uses, PFAS polymer uses, they're used in a very wide range of products. We look at mechanical components such as gaskets, seals, insulators. These are used often in aviation, aerospace, automotive industries. We also have reaction vessels and stir bars. Those are biotech and pharma industries. Membranes and coatings, polymer coatings are typical in building construction materials. Cable and wiring, also used in chemical industry as barrel coatings and things like that to prevent corrosion. They also are used in adhesives and seals, caulks, paints, sealants. So these are also common building materials. But on a health standpoint, they're also used in cosmetics and personal care products, electronics, so insulators and printed circuit boards, films, coatings for solar panels, fuel cells, lubrication products, as we mentioned, the surfactant and the repellent properties, firefighting equipment and protective clothing. So this is going to be on the safety side. Those are very common products for firefighting gear. Tray liners, non-stick coatings. So we talk about food packaging, textile treatment, so upholstery, carpet, clothing. And then again, on the health side, you have surgical patches, cardiographs, implants. These are very inert polymers, so they don't degrade. And so they're very ideal for health situations, like for implants in the body. Paper coatings for water repellency, and then waxes, such as in sporting equipment and strings for musical instruments, actually. Then when we talk about non-polar polymer uses, there's some pretty similar uses, but then there's also some very specific ones. So you have hydraulic fluid additives. This is often in aviation, aerospace, textile treatment again. So this is going to be surface treatment in like automotive, the seats. Plant growth regulators and herbicides, the short chain sulfonamides and the FOSA are common for these applications. Ultrafiltration and microporous membranes. Again, paints and coatings for building materials. They're also actually used in polymer production for binders and stabilizers. Some of them are monomers for the fluoropolymers that are produced. Again, cosmetics, shampoos, nail polish, eye makeup. So these are modern uses. Flame retardant resins, fire suppression and corrosion. So you think about AFFF use for that one. Fuel cells and battery electrolytes, water repellent coatings again, vapor suppression for flammable liquids, textiles and coatings. So again, upholstery, carpet, floor finishes, dry cleaning, but also you think about outdoor gear that's water repellent that has a lot of PFAS compounds used for those. X-ray film, medical fabrics, burn wound care, so health again. They're used as wetting agents, and then adhesive resin for wood particle boards is also a common use. So just focusing on the US right now, these are PFAS contaminated sites. So on the left is known contaminated sites. These images are from the PFAS Exchange website. So known contaminated sites, with different ranges of PFAS levels. But then on the right, we have what we call the presumptive contaminated sites. So these include areas of industrial facilities, wastewater treatment plants that might be producing biosolids, military sites such as Air Force bases, and then major airports where large quantities of like AFFF might be used and things like that. So then when we talk about PFAS sources and exposure cycles. So obviously we have the industrial input, you have some groundwater releases, wastewater discharge, wastewater that goes to treatment plants, but then you have the post-production releases as well. So you have commercial products from households that go to landfills that are in households that can cause human exposure. You also have biosolids from wastewater treatment plants that might be agricultural applied. And so then you can get food, contaminated food products, as well as environmental release from leaching from agricultural lands. So there's many different kind of sources that humans can be exposed to. The main sources that we mention typically are drinking water, food, and then dust from households where the commercial products are in use. So PFAS health effects. So a lot of the adverse health effects observed in laboratory animals through the toxicity testing that's been done involve the activation of the peroxisome proliferator activated receptor, or the PPAR alpha. This mediates actually a broad range of biological responses and possible effects. Mainly, it inserts itself into kind of fatty acid metabolism because it looks so much similar to a fatty acid. What we see in rats and mice, they seem to be the most sensitive species, with humans being less responsive in terms of the PPAR receptor. However, the PPAR dependent mechanisms associated with toxicity include hepatocellular hypertrophy, alterations in lipid metabolism, decreased pup survival. So again, this is focused on laboratory animals, and then also some immune effects. But some of the independent mechanisms also can affect liver and immune toxicity. So lab animal models have observed liver effects, developmental effects, immunological effects, and cancer effects as well. From a human standpoint, exposed populations generally fall into three categories. You have your occupational workers, which are thought to be your highest exposure, and then communities with industrial impacts are your mid-level exposures, and then the general population tend to have lower exposures. On the right is the NHANES data from different cycles, going from early 2000s to most recently 2018. And these are the average blood PFAS levels in US citizens. And as you can see, PFAS, the purple line, is generally going down over time. Some of the other compounds, such as PFNA, PFHXS, they're remaining relatively stable over the last couple decades. Some of the epidemiological studies that have been conducted suggest associations between PFAS exposure and health outcomes, which include increase in serum lipids, mainly with total cholesterol and low density lipoprotein, increase in serum hepatic enzymes, a decreased antibody response to vaccines. However, these cause and effect relationships have not been established as of yet. The International Agency for Research on Cancer and the EPA have listed PFOA as possibly carcinogenic to humans. Along with this, EPA also lists PFAS, and these are based on suggestive evidence of carcinogenic potential. So going back to the PFAS source exposure, what you're going to hear today is topics on environmental fate and transport, and then remediation of the contaminated areas, whether it's agricultural land or water, and then some of the advances that have been made for remediation purposes. And I just want to say thank you for listening.
Thank you so much, Sarah. This is excellent introduction to your topic. So let us continue with Dr. Linda Lee. She holds many roles at Purdue University in the United States. She's a professor in agronomy. She's professor in environmental ecological engineering. Today she's going to tell us more now about environmental fate, transformations, and transport of these PFAS materials.
Thank you. Glad to join you guys today. I've been working on PFAS since about 2006 in various ways. So today I'm just going to highlight some fate and transport mechanisms with a little bit of data, just to give you a flavor of what happens out there. as Sarah explained, with, you know, the PFAS is a sea of chemicals, and even to a chemist, it's a lot of chemicals, you know, over 11,000, 12,000, depending on whatever the last article is you read, of PFAS out in the environment. But once they're discharged to land, whether it's through wastewater discharge, land application of biosolids, the AFFF foams that Sarah mentioned getting used to fight fires or train, we then undergo various PFAS fate mechanisms. They're very dependent on the chemical properties. So she talked about chain length, and there, I was trying to get rid of some things here that popped over on my screen. Chain length and functional group, she highlighted those very quickly. You could spend a whole semester of class learning about those. And then various soil properties. And what we know happens, PFAS interact with the solid particles, solid phase adsorption, which many of you are probably aware of. But then it also can interact with the air-water interface, and it undergoes biotransformation. Then it can be taken up by plants, it can be run off into waterways, it can leach into the groundwater. And in many of our agricultural sites in the United States, we have something that's referred to tile drainage, which is basically a tile drain a meter below the soil surface so that we can actually grow crops. Otherwise, our soils would be flooded. And so you can have tile drain discharge. And I'm going to highlight each of those. And then you have different climatic conditions. So where you live and PFAS fate, how deep it gets into groundwater and whether it gets into your water sources and even your plants is very climate dependent. Growing degree, days, and rainfall intensity. So when we're talking about sorption, there's actually a lot of mechanisms. As Sarah pointed out, there's a lot of different types of PFAS groupings, and within each group, they have different chain lengths. But oftentimes what controls their sorption to soil is the chain length. So longer chain lengths tend to be more hydrophobic, and the polar heads. So if they have a negative charge on them, they might not be very interactive with, they'll interact different than if they have a cationic polar group. They can go to exchange sites on soil. We know both in the body and in the soil, they can have a high affinity for proteins. That's what makes them different than a lot of the organics we've worked on in the past. They don't accumulate in our fatty tissues, they accumulate in where proteins are, so blood and liver and kidney. And we know because of the air-water interfacial sorption, they can also, soil texture will matter. And then there's a lot of solution properties that can affect them, similar to a lot of organics, pH, ionic strength, and ionic composition, and dissolved organic matter. So, you know, when we talk about PFAS, we talk about parts per trillion in water and parts per billion in soils, solids, biota. And why is that? I'm just showing you one example here, where I'm showing you an organic carbon normalized sorption coefficient on the log scale versus the fluorinated carbon chain length. So we talked about longer chain lengths are more sorptive, and you see that in this trend. And this was data from both biosolids and city compost waste, you know, the trees and the leaves that get chopped up in our cities. And so we see a very strong correlation with a lot of high carbon amendments. We see a definite correlation with carbon, and we don't have to consider that many other properties of the material. That's not true in soils. When you try to describe what happens across the nation in soils, you cannot just look at carbon. Globally, it will depend on soil type. And Sarah mentioned, you know, bioaccumulation and toxicity. So again, you see this chain length effect where you see longer chain lengths within a subgroup, higher sorption. We also, or higher bioaccumulation factor. We also see that the sulfonic acids like PFAS that you hear about for the same chain length can actually be taken up even more by biota, whether it's plants or fish, whatever that is. So that's why we talk about PPT in water, because 1 PPT in water can lead to 1,000 parts per billion in a biota. So when we talk about what happens after land application, or when sewage or leachate end up in our industrial wastes, end up in our wastewater treatment plants, you get biotransformation. PFAS in the natural environment is really all about, it's a case of multiplication. So I'm just showing you 2 examples here. One class of PFAS, based on how they're produced, are telomer-based compounds, which means they have a CH2 linkage. in between a polar functional group and a tail. In this case, I'm showing you one with 2 tails, very commonly used in paper products. Or we can have our electrochemically derived PFAS that 3M initially came up with the manufacturing process, and that's unique to have this SO2 group that's linked to things. And so what happens once these are land applied or once they enter our treatment plants, they will undergo multiple steps and pathways at different rates through a myriad of intermediates to end up with what we call the terminal acids. So for the telomer-based compounds, they end up being perfluorocarboxylic acids, with the chain length that dominates being the chain length that was initially present, but it also produces shorter chain length. It's an interesting phenomena. And then for the electrochemically derived, they end up with the same chain length they started with, but then you end up with the SO3 group, so you have your PFAS. So if it was only 6 carbons with perfluorinated, you'd end up with a perfluorosulfonic acid of 6 carbons. And our problem is that these terminal products are actually more mobile than the precursors, and this is how they manage to move into our groundwater or into our air. I'm going to show you just snapshots now of some of the things I've highlighted. So here's a site that actually was a dedicated land disposal site, so where biosolids were used not for fertilizer, but just as a dedicated land disposal scenario. And in that scenario, because of 40 years of applying annually biosolids with high carbon, you actually built up a lot of carbon in the soil. And so what you see here, I'm now just putting things in classes because you don't want to see 50,000 different colors. So these grays are either precursors or intermediates, and all the other colors are those terminal compounds. And you can see here that what's hanging out in the upper 2 feet of soil are all these long chain sulfonic acids and carboxylic acids, and only the short chains are mobilizing. If we look at it a different way, this is what was in the biosolids that was land applied. You can see most of it is precursors. The orange and blue are the terminal acids. And then what do you see with depth? If we look at distribution, you see mostly long chains hanging out in the top 2 feet, some in the next 2 feet, but what you see is the blue is mobilization of the short-term compounds, short chain compounds. So what we see is once you land apply, many of the precursors break down. The telomere compounds break down very, very quickly. We see very little of them in 1 year, after 1 year. Some of the electrochemically derived are much slower degraders. You see long chain retained in the upper soil profile, mostly short chain getting into the groundwater. And interesting at this site, probably of the combination of the high carbon and the biosolids, only the 8 carbon PFOA that we've heard a lot about ever exceeded the EPA maximum concentration levels. So that was very interesting after having this high of PFAS in the soil. I'm going to show you 2 examples of where we've applied biosolids for fertilizer purposes. This is on an East Coast farm, and it had 3 different soil types. So what you're seeing here again is the orange and yellow. Those are the very long chain compounds, and then you see the blues and the grays are shorter chain, and then that's about all you see. So because our precursors have transformed. And then you also see going from a fine textured silt loam to a medium textured loam to a very large textured sandy loam, you see the difference in mobility, where the finer textured soils were retaining PFAS and the shorter chains are mobilizing. Here's another Midwest, this is a Midwest farm, also under 4 decades of land application for fertilizer. And I don't show you the distribution of PFAS, but you can see even 40 years, you're still getting all your PFAS mostly in this upper profile. And another interesting thing that we see is what we call desorption hysteresis. So if you were looking at soil concentration versus solution concentration at equilibrium, you might see something like this dashed line. But when we take this field soil and we force desorption to happen on it, it's very hysteretic, which means there's a lot of these long chain compounds that are deeply penetrated into the soil particles, and they're not going to move very long. What that means is they're with us for a long time, but it also means their mobility to groundwater is slower than we might predict. So we have all this longevity in the soil. Once we land apply, I already showed you how much of the long chains sit up in the top soil, so we can have runoff with heavy, intense rain events. And here's an example of what we had on one of our East Coast sites, where we're looking at flow rate versus time and total PFAS load in nanograms per minute. And you can see with our high intense rain events and high flow, we get high PFAS loads. What does that load look like? Well, probably shouldn't surprise you, mostly short chain compounds, mostly long chain compounds, because the short chains have mostly mobilized to groundwater and the long chains are left in that upper soil profile, so subject to runoff for several years. And this is actually after this data here is when there had not been land application for 7 years at this site. And what does that mean? Where does the runoff go? Well, it can go into a lot of places. We've been looking at ponds in some of our sites. And so this is just a little snapshot, not to be scary, but just to show you the bioaccumulation and why we talk about PPBs in biota. So what we see here is a lot of PFAS in these soils, in these fish and the different biota, very little that you actually would detect in the water and sediment. The water's on a PPT level, but the accumulation of PFAS in the largemouth bass and the bluegill was quite substantial. And this field has not had biosolids on it for over a decade or more. And so it's just showing you the longevity in the soil, longevity once it gets into the environment. And so this can be a concern for grazing cattle, people that fish in the ponds, obviously, and then exposure to humans. I want to show you another scenario with when you have tile drainage. So a lot of our ag fields in Indiana and a lot of the Midwest and even on the East Coast are tile drained. And this is that pipe I was saying. It's a perforated pipe about 1 meter below where you're growing your crops. And so we did, the county I live in, we did some very intense hundreds of water samples, rural water and surface water. And first, what we saw in the rural water supplies, even though all these green that you see is where land application of biosolids happens, everything was very low in our rural water, and nothing was above EPA's drinking water limits. So for those of you that don't know, PFOA and PFAS are regulated at 4 parts per trillion. And then when we looked at the surface water, we expected to see much higher concentrations because the hypothesis was, yep, it's going to the tile drain and exiting to our ditches and streams, and it's not migrating to groundwater because of that. And so, yes, we had much higher detection, much higher PFAS concentration, and the number of PFAS we detected in streams was much higher. And with the highest one we saw in our area of close to 100 ppt of the C4 sulfonic acid, which has replaced PFAS. So lastly, let's just highlight a little bit of plant uptake. So yes, PFAS will sorbed to roots and they can be taken up by plant roots and then into the plant primarily through transpiration. PFAS uptake will increase as PFAS soil concentrations increase. The more mobile short chains, they're what dominate in the pore water, and that is what is going to be mostly transpired and what will dominate in the above ground matter of the plants. So that's kind of good because we think that you need more of a short chain to be adversely impacted, but as Sarah pointed out, we still have a lot of unknowns in the adverse impacts. The highest uptake is going to be under any scenario where you're promoting transpiration. And so that would be plant type and species, the soil type you have, how much pore water is held there for the plants, and the weather patterns. So any kind of scenario where you're really keeping PFAS in the pore water and you have a lot of sun degree days and plants that need a lot of water, you're going to see a lot more PFAS uptake. You will get sorption to root vegetables and then diffusion into the vegetable, which can include the long chain PFAS. You see more short chain in the above ground mass, but the longer chain PFAS can sorb to the skin and diffuse into our vegetables. For PFAS that enter the seed, it's complicated. So what we are starting to see now is that a lot of PFAS aren't entering the seeds of things. For example, we did a soybean study on very, very contaminated soil, and we saw only the 3 shortest chain, not ultra short. I don't think Sarah talked about ultra short, but ultra short are things smaller than C4. But we did see C4, C5, C6 in the soybean, but the soil had about 1,800 parts per billion PFOS, and no PFOS showed up in the bean. It did show up in the husks, and it showed up in the leaves. And just like in corn, we see a lot in the leaves and the stalk that's used for silage, for feed, for plant, for cattle, but we don't see very much at all in the cob or in the corn. We do know, because of, we hypothesize that there is non-specific substrate transporter that lead to the smaller short chains that look like little fatty acids or kind of mimic small amino acids that allow them to get into the actual seed, plant seed. But other than that, you don't see a lot in the fruit or the seeds. It's mostly that leafy above ground mass. You will see variations between cultivars. We know spinaches and lettuces can take up a lot of PFAS, but different cultivars will vary. And we know that they can be correlated to root proteins and lipid content. I think nobody eats that much in one day of any particular plant. So it seems like some of our biggest challenges are really PFAS uptake into field crops like our grasses, our silage that gets fed to our dairy and livestock. And this is what leads to PFAS being in unacceptable levels in our meat and our milk. And so there's a lot of things we're all working on now to try to mitigate that. And I know, I think Clinton is.
Clinton Williams, who is at USDA Arid Land Agricultural Research Service. He's working there on water management, and conservation research unit. So Clinton now is going to transition into mitigation, remediation, and risks of these materials. Go ahead, Clinton.
Thanks. So I'm Clinton Williams. I'm located in Maricopa, Arizona, which is just a little bit south of Phoenix, Arizona. And I'm here to talk about mitigation, remediation, and risks, and the risks that we face from PFAS. So just to start with, looking at mitigation, in essence, how can we limit how the PFAS moves within the environment? And the quick things we're going to talk about is adsorption. Linda talked a lot about that, but we'll show some ways that we can sort of remove PFAS from places, membrane filtration, phytoremediation, things like biochar, and then some of the newer technologies are looking to try and collect PFAS at that air-water interface. So to start with adsorption, it's been shown that PFAS can be absorbed to granulated activated carbon. It's cost effective. It's been shown to be very effective. To get to really low concentrations, we're not quite sure about getting all the way down to the 4 parts per trillion drinking water standards, but that's sort of the direction we're moving in water treatment. And then there's also some opportunities to get electrostatic interactions from for short chains. We can use ion exchange that uses pH-dependent charges from those carboxylic and sulfonic acid ends that can be sorbed onto anion exchange resins. The resins are this means that we also have pH-dependent charges to remove the PFAS from water, as well as we can use both cation and anion exchange resins. And the anion exchange resins are much more effective for the long chain PFASs, and they're fairly cost effective. Other ways we can remove PFAS from specifically water, and the adsorption and the ion exchange resins and membrane filtration are all sort of from water situations. We have anywhere from microfiltration all the way down to reverse osmosis, and as we go from microfiltration down to reverse osmosis, this is sort of the size of, as you go down this, the pore sizes decrease. So we go from conventional filtration, we have microfiltration all the way to reverse osmosis, where the particle sizes we're removing are anywhere from, say, a micron down to 10 to the minus 4. And the other thing to think about this is that as you go to smaller and smaller pore sizes, we're removing more and more things from the water. But as you do go to smaller and smaller pore sizes, we're also increasing the pressure that's required to remove these compounds from the water. There's high efficiency with the membrane filtration, but it's also one of the problems with it is we have a high concentration of PFAS in the permeate, or I mean in the concentrate. We talked about phytoremediation. So this is a way where if you have soils, you can remove the compound, the contaminant from the soil. As you have the compound in the soil, as Linda talked about, we can, wherever the water moves, you can concentrate the compound in, say, the leafy portions of the soil or of the plant. So as you take the compound up, it's going to collect within the plant. Once that happens, you can take the plant, remove it from the soil, and as you do, you've actually removed some of the PFAS from the soil, from the place where it's contaminating. The use of biochars, we can use biochars to immobilize the PFAS in soil. So biochars are relatively inexpensive. They're somewhat similar to GAC, but they're not as activated or don't have as high of absorption capacity. But they are things that you can just add to soil. So if you have a soil where you have PFAS contamination, adding GAC to the soil can help to remediate or to immobilize the PFAS so it won't move to the crop you're producing. So this is one of those things where if you can amend the soil with high enough amounts of biochar, we might be able to immobilize the PFAS before it gets to the plants that we're growing. Some other newer things that are being looked at to understand or to remove PFAS from water, things like using micro and nanobubbles in dissolved air flotation, so that you have a water coming in that has the contaminant in it, and then you introduce very small bubbles that can then start to join together, get bigger and bigger and bigger, and as they do, you form an air-water interface where the contaminant can join with that air-water interface and then be floated to the surface. One of the advantages to this is that as the bubbles start to form, they rapidly rise to the surface, and you can then skim them off the top to remove any of the PFAS. Again, as you do that, though, the PFAS has just been moved from this water, so you have clean water coming out, but the water that comes off the top that that's undergone dissolved air flotation now has high PFAS contamination. So again, with the mitigation, we've figured out how to immobilize the PFAS or to sort of remove it, but we've now concentrated it into another compartment, whether it's foam off the top or in a concentrate from a filtration or onto biochar or GAC. The thing about that is, though, the PFAS is still there. So that comes down to remediation. So in essence, actually destroying the PFAS. And there are a few potentially useful treatment technologies we have that have come about, some hydrothermal treatments, mechanochemical, thermal destruction, doing some advanced oxidation and photocatalysis, as well as just now biotransformations are starting to come, and that's what Jude will talk about afterwards. So hydrothermal treatments, usually this is using subcritical or supercritical water, mixing in various sorts of like either catalysts or different reactants that can work within the system. And there's been movement on, and it's been seen that we can start to get PFAS removal or degradation. The issue again with this is if we just take it from a longer chain molecule to a shorter chain PFAS, as Linda was talking about, we still have PFAS as a problem. So all of these need to go all the way to complete destruction. One promising treatment that's come out recently, a lot of the soils that are contaminated with PFAS are contaminated due to AFFF, which is firefighting foam. And it's been shown that you can actually take contaminated soil, mix it with some stainless steel grinding beads, put it in horizontal ball mills, and actually get complete destruction of the PFAS. It's shown that it can happen. One of the problems with it is the amount of time it's going to take, and to actually treat large quantities of soil is going to take a long time. So right now the direction is to try and find, are there things we can add to help increase the PFAS destruction by lowering the energy required to do that. Other things, thermal destruction. So we can do incineration, induction heating, focused microwaves. These are all methods to where we're trying to increase temperature to destroy the PFAS. Some of the temperatures needed to actually break that carbon-fluorine bond are up around 200 or 2000 C, which means that we're in a situation where you really got to get high temperatures. A lot of times in incineration, we don't have long enough retention times to get complete destruction. So currently, DOE and DOD do not look at commercial incineration as a top or a way to destroy PFAS, because first off, we can't measure the PFAS that's released after the incineration, and the residence times that are needed are oftentimes too long for the way the incinerators are set up. Typically, an incinerator will have, less than a second retention time. If you're lucky, you can get a few seconds retention time, and we need to increase those residence times to actually get complete destruction of the PFAS, that fluorine carbon bond. There's been some look into advanced oxidation. You could use electrochemical oxidation. You can have UV induced or UV accelerated oxidation, mixing it with hydrogen peroxide and other different sorts of oxidants. The issue with this is it's still sort of in its early stages, trying to figure out how we can get this, and then also, are we doing simply partial degradation, or are we getting complete mineralization and breaking all those carbon-fluorine bonds? So that's what we need to really look at, because oftentimes with advanced oxidation, you're just getting a transformation to something that's maybe more or less toxic, but still it's not actually removing it. The last thing to talk about with mitigation, or with like actually destroying the compound, is biotransformations. This is something that's just now becoming more and more common. Jude will talk a lot about this, but I just want to show one situation where they've actually been able to find a microorganism that can destroy the PFAS, the PFAS completely, and they're actually showing that by getting fluoride production, as well as measuring some of the byproducts to show that yes, some go up, but then they come back down, and you do get some the fluoride is actually being produced. So that's sort of an interesting future as far as expense and like being able to actually use these technologies. Biotransformation probably has the most promise for being able to do it inexpensively, but it's also probably the one that's the hardest to actually get to work. So the risks associated with this is, Linda talked about, we have biosolids. In the United States, there are a number of plants that produce more than 5 million grams of biosolids a day. We also have a number that are producing more than 50 million grams of biosolids a day. And all those biosolids, for the most part, have been land applied, because for the longest time we've seen biosolids as a good fertilizer source. So the other thing to look at with the risks are the fact that we have numbers that are the risk assessment numbers that are coming out for biosolids, or for like water and how it's affecting people. And this is just a study that was done recently where they took and compared all the different standards that were associated with PFAS, like maximum daily limits and the risk assessments associated with it. And if you see here, there's a 5 order of magnitude difference between the different outcomes of the different studies that have come up with risk assessment. And really what it comes down to is it all depends on what they assume is their safe dose, as well as how many uncertainty factors they include. And this really has come up with, on the low end, we have the 4 part per trillion number that EPA has come up with, as well as we have higher numbers for, say, New Zealand and Australia, which one of the problems with this is using the exact same data, the exact same papers, all these people came up with these vastly different numbers, which means that we need to have maybe a better understanding or some better controls on how we do our risk assessment to get some uniformity to these risk assessments so that we can actually get people to have more faith and confidence in the numbers. Just some things to look at also. We look at some water samples that were taken throughout the state of North Carolina, and you can see that there are some places with very little PFAS, some with much higher PFAS concentrations. So it kind of depends on where you come from, what you're like, what's around, whether there's biosolids applications, things like that, to determine what the different concentrations in the water surface waters might be. Some other quick just data to look at, and this is looking at the absorption of different PFASs onto microplastics. And if you look here along the bottom, these are different kinds of microplastics, and these are actually weathered microplastics, not virgin microplastics, and shows that polyacrylamide can have some fairly high absorption of these different PFASs, and it's also pH dependent, which you'd expect based on those sulfonic and carboxylic acid heads. The other thing about that's a problem is if you have microplastics that move from, they sorb in one place, that's the bars, and then they desorb in a different place. As the pH changes, you can actually push much more of the PFAS off of the microplastics. So that becomes an issue when we look at how it might move through the environment and what other co-contaminants might be around. Again, if we looked at just soils where we had some organic amendments applied and soils without organic amendments, and these were biosolids as well as manures. If you apply biosolids, you can end up, or manures, you can end up with much higher PFAS concentrations. And the final thing I wanted to show is just that the cost of destroying these PFASs in the environment is huge. A paper that was done that estimates the cost to destroy all the PFAS produced annually is between 20 and 7,000 trillion dollars, with global GDP being 106 trillion. So unless we can get microbes that can do it for cheaper, we're in a situation where the PFAS that we're producing that is necessary for things like microchip processor production, as well as protecting firefighters and firefighter foams, all those things are necessary. We don't have real good alternatives right now, and the cost to the world is going to be very high if we can't figure out ways to mitigate, remove, and destroy these cheaper. With that, thank you. And
well, thank you, Clinton, so much. Thank you so much. So with that, let's transition to the last speaker, Jude Moll, who is with Sustainable Agricultural Systems Laboratory in Beltsville, Maryland, with USDA. Now, Jude brings a very interesting angle where his research really is focusing on the molecular ecology at the plant soil water interface, and in the context of plant health, nutrient acquisition, and productivity. So his research sort of contributes to soil health management and various recommendations. So with that, Jude, go ahead and close our webinar. Go ahead.
Okay. Thank you very much, and thank you to all my colleagues who spoke before. I'll try to be quick here. Everyone gave a great introduction. I'm going to give a perspective on the biotechnological approach to PFAS remediation, and really, instead of give a lot of examples, kind of outline sort of an approach or a mindset to go about taking a biotechnological approach. Let's see. Okay, so PFAS is it's a biological problem of anthropogenic creation, and so it makes sense in some way.
Okay,
Now,
go ahead.
okay,
Yep,
all
go
right,
ahead.
I'm sorry about that. Okay, so taking a technological approach makes sense because these are novel compounds in nature, and they're although they're novel, there's many examples of biological catabolism in nature and the lab, and so there's a need to investigate where PFAS interferes with biological processes. These points might be critical bottlenecks that give us clues on how PFAS is interacting with these systems, and may give us insight into how to remediate. And in some ways, we can think of the concept in the art of war, where using your enemy's strength to your advantage, and it's almost like we have to take that approach to understanding how to address these compounds. My slide is not advancing. Okay. So as folks outlined previously, there's a variety of physical and biological pathways for PFAS degradation, and you can see here a list of things that, as was pointed out earlier, in many cases result in depolymerization or reduction in molecular mass, but in many cases not complete degradation of these compounds. And so the thought is that most likely we'll have to use a combination of approaches, as Clinton just pointed out in the very last few slides, the use of nanobubbles or foams, and we could envision an approach where potentially a combination of physical manipulation of the contaminated material plus biological seeding and or manipulation of these films might be a path forward. And I have down here a few references to reviews that point out these concepts in a little more detail. So there is growing evidence of direct biological metabolism and or catabolism of PFAS compounds. This has been observed both anaerobically and aerobically, and both as individual isolates that have a function that break down all or part of PFAS molecule, to consortia of microbes that might cross-feed, where one species or serotype has a particular function, and then the byproduct of that metabolism is then the substrate for a following step by another microbe. And so there may be examples where we may have to culture specific consortia of bacteria that carry out multiple processes that result in the degradation of PFAS compounds. So I'll just point out here two major differences in the classes of enzymes that associate from a catabolic sense with PFAS compounds are transporters and dehalogenases. And so these haloalkene dehalogenases are the enzymes that physically break the bonds in the PFAS molecules, and as Clinton and others alluded to, where you have sequential degradation of these compounds, whereas in this case a fluoride ion transporter, but there's also other transporters that can bind and or move PFAS molecules. And so when we're thinking about remediation, I think we have to really think outside of the box, and that in some cases it may not be optimal to just focus on degrading the compound, but in some sense is isolating it or concentrating it in a particular location or onto a film for further processing might be the most economically feasible approach to doing this. And so really kind of the main theme here is we're going to have to take multiple methods and probably a combination of biological and physical approaches to remediation. So there's examples of, as I mentioned, individual strains that have been shown to metabolize PFAS, and in many cases this is not to full degradation of these compounds. And you can see these have been observed both as individual isolates, but then also as mixed cultures. And probably the best review to date on these processes within microbial communities is from Samranda et al. And they identify pseudomonads and some other cytomicrobium as key genera that have been identified or associated with the degradation of these compounds. And so some microbiologists may chuckle and say, oh, pseudomonads, break this down, because they're a very broad group. So clearly we're going to have to dig into the actual functional genomics of these different isolates to really understand the mechanisms that are responsible for PFAS degradation. So getting back to this theme of understanding the nature of your enemy. So I just wanted to look and I started thinking about how and where PFAS ends up in plants and or animals. And so if you just look at things that can cross the blood-brain barrier, and there's really sort of well-known or well-defined classes of compounds that can get through there. And taking that approach of understanding the fate, as Linda pointed out, the fate and the transport of these molecules may give us insight into not only how we might address specific illnesses or impacts of this, but it may give us insight into the biological mechanisms that are necessary to get those compounds there. So although they may concentrate in different parts of our bodies or plants, if we delve into the molecular mechanisms of these processes, it may give us insight into how or strategies for bioremediation. So here's an example of differentiation of PFAS constituents in different parts of the body. And as Sarah pointed out, in many cases, when PFAS moves into an organ like the liver, it induces a number of genetic responses. So that's the response of the organism to the PFAS present there, but it's being recognized or sensed in some way in that process, and getting at those mechanisms might be a key to a biotechnological approach. So an example of that is here. In many cases, PFAS and PCBs, for example, interfere with synaptic neuron messaging and control, and specifically in the development of organs in children. And the way this occurs is with this rhododione receptor, PFASs and PCBs can bind to the actual receptor that allows calcium to move out of the synaptic junction. And this binding is the problem. It's the cause of this neurogenic malfunction in humans. But if we look into the mechanism, and this is just an example, if we look into the actual binding, the kinetics of why these molecules, how they bind, where they bind, we may not actually be able to solve this problem, but we may be able to use that knowledge in some other application. And here's just a hypothetical application of that. So for example, there's a technology that exists now, and you may be familiar with it in England and the UK, Oxford nanopore sequencing technology, in which a protein, it's a hybrid membrane protein chip, and it can recognize and funnel DNA through this protein channel, and in that process sequence DNA. It's a great advance, and it utilizes both physical a silica chip and protein engineering to carry out this process. I can envision, with better understanding of the ways PFAS binds to proteins and how cells move PFAS in and out of the cells and how bioaccumulation is occurring, we may be able to develop something like a membrane that can specifically, with high affinity, bind PFAS and move it to the other side of the membrane, acting as a step of concentrating, preferably in some sort of unit that then could be disposed of. So I went through that pretty fast, but in conclusion, we know PFAS are toxic, they bioaccumulate, and their persistence is recalcitrantry is due to the way these molecules interact with the kinetic function of our biological systems, and understanding these kinetics will be crucial to progress in the field. And these physiochemical properties of PFAS are relevant to biodegradation and bioaccumulation, and these may give us clues to the mechanisms for removal or remediation. And a comprehensive understanding of transformation and defluorination pathways in microbes will be critical to identify pathways that may be used for transformation into other systems for bioremediation and increased efficiency in this. And I think there's a great opportunity here for machine learning and artificial intelligence tools to help us understand some of these pathways and look at interactions that we may not foresee through typical models of protein-protein interaction or protein molecule interactions. So with that, I'd like to thank you and turn it back to our
I'm going to jump in on behalf of Jonas. I am the Assistant Publisher for Sustainability Science and Technology. I appreciate we have run a little bit over with the webinar. First of all, just wanted to thank each and every one of you for attending today and showing great interest, and to all of our panelists. It was a really, really interesting discussion. I've definitely learned quite a few things from it as well. We did promise some time at the end for questions, but unfortunately we have run a bit over. So if any of you did answer any questions, we have made a note of them and who's asked them, and we will get back in touch with you via email to provide you with an answer on that. And if any of your colleagues have had to run off a wee bit early, you can let them know that the recording will be sent out to everybody who was registered, so they can always catch up on any of the bits that they might have missed. But for now, we just want to thank you again and encourage you to check out the journal. If you have any thoughts or want to reach out at all, we're always welcome any communications from you guys. And yeah, so once again, just thank you everybody for coming, and to all of our wonderful panelists, and of course to Jonas for being our chair today. And wishing you all a good day wherever you may be. Thank you.